Thank you so much for that nice introduction. And I'm sorry I couldn't be there with you all, but I'm happy to at least to be able to participate by Zoom. So let me just see if I can share my screen. Can everybody see the slide deck? I'm going to assume you can see them.
Yep.
So
I think well.
Okay, great. So I'm going to take a little bit different approach. You've heard some amazing, beautiful science in these initial talks, and I'm going to talk about how we're trying to take all the great science being done in the aging field and apply it to humans in clinical trials. So my company, Tornado Therapeutics, is developing best-in-class next-generation rapalogs. So rapalogs are derivatives of the oral mTOR inhibitor rapamycin that many of you know about. The rapalogs are FDA-approved drugs for the treatment of multiple diseases, including cancer. But why they're important to the aging field is that rapalogs probably have additional untapped therapeutic potential because this one drug, rapamycin, is the best validated therapeutic to extend lifespan and health span in model organisms. And this raises the question, could rapalogs or rapamycin actually have benefit for human aging? So to begin to address this, we have in-licensed a portfolio of rapamycin derivatives from Novartis that are predicted to be best-in-class compounds with enhanced safety and or efficacy as compared to first-generation rapalogs like rapamycin, and we're moving these into the clinic in the first half of 2024. But just to step back again, what is mTOR? So mTOR is a protein kinase that's the key link of linking the availability of nutrients to growth and reproduction. So when organisms eat, this activates mTOR, and when mTOR is activated, it upregulates growth pathways like protein and nucleotide and lipid synthesis that cells and organisms need to grow and reproduce. Then during periods of fasting, mTOR is inhibited, and when mTOR is inhibited, protective pathways are upregulated. And these protective pathways that are upregulated are not only important for surviving periods of famine, but they're also associated with increased lifespan. So normally when we're young, mTOR sort of has a circadian rhythm. You can see in young mice, this is looking at mTOR activity in the liver as assessed by levels of phospho-S6. mTOR is activated when the mice eat. This is upregulating protein and lipid and nucleotide synthesis so cells can divide. And then when mice are in periods of fasting, mTOR is inhibited and these protective pathways that are upregulated that are important for cell health. And one of the reasons fasting and intermittent fasting may have benefits is because it causes inhibition of mTOR. The problem, and this has been shown now in both human tissues and in mouse tissues, mTOR may become hyperactive as we age. And in certain mouse tissues, including liver and muscle, it's been shown that mTOR in old mice stops responding to fasting and just stays on all the time. So there's no periods of the day when mTOR is inhibited and protective pathways are upregulated. And what this suggests is just turning mTOR down to young levels or even just intermittently inhibiting it may have benefits for aging-related conditions. So one of the ways to inhibit mTOR is with this drug that I mentioned called rapamycin that has an interesting backstory. So there's water in a crater of a volcano on a remote island in the Pacific Ocean called Easter Island or Rapa Nui. And it was reported that this water in this volcanic crater had some sort of health benefits by local residents. An investigator studied samples of the soil in this volcanic crater and found that it contained a bacteria that produced a molecule which they called rapamycin after the name of the island. And it was subsequently found to be a very specific and potent inhibitor of mTOR. Since discovery of that compound, it has been shown by multiple groups that this one drug can extend lifespan and health span in every organism studied to date: yeast, worms, flies, and mice. So whether you use rapamycin to inhibit mTOR or whether you genetically inhibit mTOR in these organisms, lifespan and health span is extended. And since these organisms span 1 billion years of evolution, it's likely that mTOR inhibitors and rapalogs in particular, the particular class of mTOR inhibitors, are going to have benefits for human aging. What's interesting about rapamycin is that even if it started late in life in mice, it still extends lifespan. So the study on the left is a famous study done by the Interventions Testing Program at the National Institutes of Aging in the US, in which they took 3 different sites across the US and treated genetically heterogeneous mice with different compounds to see which compounds across these 3 different sites significantly extended lifespan. And the first compound where they saw robust effects was rapamycin. And you can see even when it was started at 600 days, when the mice were 600 days old, which is the equivalent of about a 6-year-old human, that was sufficient to significantly extend lifespan, as you can see in the red line, as compared to the control animals in black. And Dudley Lamming subsequently showed is even if you only give rapamycin once every 5 days, starting when mice are already 600 days of age, that's sufficient to extend lifespan. So this is very exciting for humans because could we start mTOR inhibitors relatively late in life in humans and extend lifespan? But we've really never addressed this question, and we've done very little to determine what effect mTOR inhibitors and rapalogs in particular have in human aging. And the reason why we haven't done more is because there's been 2 main hurdles. The first hurdle is that rapamycin and its FDA-approved analogs, the rapalogs, have little or no remaining patent exclusivity, and this has diminished investor interest in spending lots of money testing them in new indications. Novartis realized this back in around 2015 and put together a big team that included their expert rapalog chemists to come up with new rapalogs that would have the patent runway for them to test in aging-related conditions. This was a tough hurdle because so many rapalog structures had already been disclosed in previous patent applications. But after many years of work, this group came up with a beautiful portfolio of new compounds. But as often happens in industry, Novartis changed its focus by the time these rapalogs were ready. They were no longer interested in aging biology as a strategic focus. So this program got orphaned and we were able to bring it into Tornado. The second hurdle that has sort of stymied people testing rapalogs in humans is that the first generation rapalogs at high doses have some side effects that are considered maybe too unsafe to use for a lot of aging-related conditions. What's exciting about this new portfolio, as I'll show you, is some of these compounds have markedly improved safety as compared to first-generation rapalogs, such as rapamycin, and that's really going to make it easier for us to test them in a variety of aging-related diseases. So if you had to design an ideal rapalog to treat aging-related conditions in humans, you would predict that it would be a rapalog that specifically inhibited mTOR in a complex called TORC1. So mTOR signals in 2 multi-protein complexes. One is called TORC1 and the other is called TORC2. It's inhibition of mTOR in the TORC1 complex that upregulates protective pathways and extends lifespan. Inhibition of mTOR in the TORC2 complex has actually been shown to decrease lifespan in mice and causes adverse events such as hyperlipidemia and hyperglycemia. So what's always thought it would be the goal is to develop a rapalog that specifically inhibits this TORC1 complex. So the first generation rapalogs like rapamycin potently inhibit mTOR in the TORC1 complex, but they also partially inhibit mTOR in the TORC2 complex. What's exciting about the Novartis portfolio is the majority of the compounds are TORC1 selective. They do not inhibit mTOR in the TORC2 complex. So that is very exciting for developing for aging-related conditions. Then about 26% have TORC1, TORC2 inhibitory activity similar to rapamycin, and 14% are what Novartis called super rapalogs, which are even more potent TORC1 and TORC2 inhibitors than rapamycin and other first-generation rapalogs, and they may have efficacy in autoimmune disease. So these are just data showing the relative TORC1 and TORC2 inhibitory activity of everolimus, which is the first-generation rapalog that Novartis markets, versus two of our next-generation rapalogs, TOR101 and TOR104. And you can see everolimus inhibits TORC1 with sub-nanomolar potency, but also partially inhibits TORC2. TOR101 is one of our TORC1 specific rapalogs. It also inhibits TORC1 with sub-nanomolar potency, but it doesn't inhibit TORC2 at all, even up to the highest dose we've tested, which is 300 times higher than the TORC1 IC50. I'm sorry, it's 1,000 times higher than the TORC1 IC50, still no TORC2 inhibition. And then TOR104 is one of the super rapalogs that's a more potent TORC1 and TORC2 inhibitor than everolimus. So I'm going to focus the rest of the talk on our lead compound, which is this TORC1 specific compound. So we tested it in vivo in rats and looked at the ability of this compound when given orally to inhibit TORC1 in a variety of organs. So you can see in rat liver, kidney, and muscle, 3 hours after giving a dose of 3, 10, or 30 mg per kg of this compound, there's beautiful TORC1 inhibition in liver, kidney, and muscle, even at the lowest dose tested. And then TORC1 activity recovers after 24 hours in a dose-dependent way. So this functions just as we would like it to as a TORC1 inhibitor. What really differentiates this compound, though, is the safety. So before you move into the clinic with a compound, you have to do preclinical good laboratory practice toxicology studies in 2 species to determine what is the lowest dose of compound where you start seeing toxicity and where do you see toxicity. So this is looking in a 28-day, what's called GLP toxicology study in rats. When they're dosed with this first-generation compound everolimus, and rapamycin looks just like this, at what dose do you start seeing tox and where? And what you can see is you start seeing thymic and lymphoid toxicity at 0.5 mg per kg. You see heart, lung, testicular, and uterine toxicity at 1.5 mg per kg, and eye and bone toxicity at 5 mg per kg. So now we've taken our lead compound, TOR101, and done these same 28-day toxicology studies in rats and comparing where do we see toxicity. So what's remarkable is this compound has dialed out almost all the toxicity of everolimus or rapamycin. The highest dose we tested in rats is 250 mg per kg. We see no thymic or lymphoid atrophy, no heart toxicity, no lung toxicity. The one toxicity that is retained is the testicular tox, because you need TORC1 to make sperm. So all TORC1 inhibitors, all rapalogs, all mTOR inhibitors will decrease spermatogenesis and sperm counts. But we've dialed out the uterine tox, the eye tox, and the bone tox. So this is a really nice profile to be able to move into the clinic. The second species that you use when you're testing toxicity of rapalogs are non-human primates, and we see this same differentiation. At the highest dose we tested in non-human primates of TOR101 of 125 mg per kg, we have seen no toxicity, whereas you see toxicity at 1.5 to 5 mg per kg with everolimus. So we're very excited about moving these compounds into the clinic for aging-related conditions in humans. So the tricky part is what indication should you choose to test these compounds? Because there are many indications where rapalogs have shown benefit either preclinically or in the clinic. And currently, our thought is we are going to test two indications where rapalogs have been shown to have benefit in the clinic, previous generations of rapalogs or mTOR inhibitors. So that's in cancer indications. Rapalogs are approved. First generation rapalogs are approved to treat cancer. We know they work in this indication. And also, we are targeting improving the function of the aging immune system as a new way to treat viral infections in older adults. And I'll talk about both of these indications. Just quickly, a potential oncology indication for our lead compound is a lymphoma called diffuse large B-cell lymphoma. This is one of the most common non-Hodgkin's lymphomas, and the standard of care for this lymphoma is a chemotherapy regimen called R-CHOP. But you can see about 70% of patients respond to R-CHOP, but 30% of patients relapse and aren't cured by this chemotherapy regimen. So Mayo Clinic in around 2015 said, if we add a mTOR inhibitor, a rapalog to R-CHOP, could we enhance the efficacy, the anti-tumor efficacy of R-CHOP? And they found it was a small trial, but the results were remarkable. This is showing event-free survival. They had a 96% event-free survival compared to the 70% that you see with R-CHOP alone. And the only event that occurred in this trial was actually a different kind of lymphoma. No one relapsed with diffuse large B-cell lymphoma. And when you look at survival, they saw a similar remarkable effect. It was 100% survival. And as of 2023, all of these patients have remained in complete remission and none have relapsed. This was a small trial. It needs to be reproduced, but we think this is a really interesting potential indication for our lead compound, particularly because it's so safe. But we wanted to make sure could this TORC1 specific inhibitor have the efficacy of the first generation rapalogs in this cancer indication? So we tested this compound in combination with R-CHOP in a model, a relatively treatment-resistant model of diffuse large B-cell lymphoma. You can see in black tumor growth in vehicle control treated animals, in green tumor growth in animals treated with a standard of care, R-CHOP, in blue tumor growth in animals treated with this very safe TORC1 inhibitor, TOR101, and in red, tumor growth in animals given a combination of R-CHOP and TOR101. So this is exciting because that combination of TOR101 and R-CHOP leads to almost complete tumor regression, just as was seen in the clinic with a first generation rapalog plus R-CHOP. So this is a potential indication for this next generation compound. But I think more interesting to this group is our efforts to use mTOR inhibitors to improve the function of the aging immune system. And I'll spend the rest of the talk on that. So we all know COVID-19 is more severe in older adults, but it turns out every viral respiratory tract infection causes more severe disease in older adults. This was a study done by the CDC prior to the COVID-19 pandemic, just looking at people of increasing age who get infected with either common coronaviruses, influenza viruses, or rhinoviruses, which are the cause of the common cold, and their rates of hospitalization. And you can see the older that people get, the more likely they are to be hospitalized when they get all these different kinds of viral respiratory tract infections. So how do we deal with this? Well, our current approach is a one bug, one drug strategy. So we develop drugs like remdesivir or Paxlovid that directly target a virus like SARS-CoV-2. But the problem is the viruses develop resistance to drugs that directly target them. And these one bug, one drug drugs don't protect older adults from all the other kinds of respiratory tract infections that are quite dangerous. So we're saying instead of targeting each individual virus, why don't we target the aging immune system? And why don't we see, can we make the antiviral immune response of older adults better so that we can protect them from all sorts of different viruses without inducing viral resistance? So what's wrong with the antiviral immune system of older adults? Well, one of the problems is they have a deficient type 1 interferon response to viruses. So this type 1 interferon response is the first line of defense against viral infections. So when cells like plasmacytoid dendritic cells detect a virus, they secrete type 1 interferons, which bind to interferon receptors on other cells and induce the expression of hundreds of different antiviral genes. But the problem as we get older is we don't have as robust an interferon response to viruses. And this has been shown by many groups. In the top right, the investigators took whole bloods from younger adults or older adults and exposed the blood to herpes simplex. And you can see the younger adults in black make significantly more interferon than the older adults shown in red. And similarly, in the bottom graph, these investigators took monocytes from older and younger adults and infected them with influenza virus. And you can see the younger adults make significantly more interferon than the older adults. So how can we boost this interferon response? Well, unexpectedly, we found that one way to do this is with low doses of mTOR inhibitors. So the data I'm showing here was from a phase 2 trial where we treated older adults for 6 weeks with very low doses of either a rapalog, a everolimus, or a different kind of catalytic site mTOR inhibitor called BEZ235. And we said, let's just do non-hypothesis-driven RNA-seq. What are the genes whose expression significantly changes in older adults who are given mTOR inhibitors? We thought we might see autophagy upregulated or mitochondrial biogenesis upregulated, but the only genes that were really, or the vast majority of genes that were significantly upregulated in older adults, 65 and older, who get low doses of mTOR inhibitors are interferon-induced antiviral genes. So you can see in this volcano plot, all of the genes that are highlighted in the white triangles, right rectangles, are interferon-induced antiviral genes that play a critical role in fighting viral infections. So we said, well, if these people are upregulating their antiviral immune response, are they having fewer respiratory tract infections, which are mainly viral, caused by viruses? And here's what we found. The people who got the catalytic inhibitor or a rapalog or a combination, they all reported having significantly fewer respiratory tract infections than people who got placebo. So we thought, great, maybe we can use low doses of mTOR inhibitors to enhance antiviral immunity in older adults and protect them from viral respiratory tract infections. We moved forward with this drug called BEZ235 because everolimus was coming off patent, so we couldn't continue to develop it. We did 2 large phase 2B and phase 3 trials to see if we could use this low doses of this mTOR inhibitor to protect older adults from viral respiratory tract infections. And here are the results of the 2 trials. If you look at just incidence of lab-confirmed respiratory tract infection, there was this 31% significant decrease in people who got the mTOR inhibitor compared to placebo in the phase 2B, but just an 11% non-significant decrease in the phase 3, although the phase 3 turned out to be underpowered because the incidence of respiratory tract infections was 50% lower in the phase 3 than the phase 2B. But when you look at the incidence of respiratory tract infections with severe symptoms, this was a pre-specified endpoint, you start seeing a bigger treatment effect in both the phase 2B and the phase 3, although again, the phase 3 wasn't significant. And if you look at the most severe infections, those that require hospitalization, again, the treatment effect in both the phase 2B and phase 3 seem to be getting bigger. So what we thought is, even though the phase 3 failed and was underpowered, we thought this suggests we didn't have the right endpoint anyway. Maybe that low doses of mTOR inhibitors and this upregulation of antiviral immunity has a bigger impact on severity of infections than just incidence of infections. So we did one final trial to test a severity endpoint with low doses of mTOR inhibitors, and we looked to see if we treat residents of nursing homes experiencing a COVID-19 outbreak with a low dose of BEZ or placebo, will we decrease the incidence of severe COVID-19? So in this trial, when there was a COVID-19 outbreak in a nursing home, people who didn't yet have COVID were either randomized to BEZ, the mTOR inhibitor, or placebo for 4 weeks, and then followed for 4 weeks. And the endpoint was FDA approved, the percent of people who developed severe COVID-19. And here's the results. It was a small trial, just a pilot trial, but 23% of people who received placebo got severe COVID-19 and half of them died. This was before vaccines were available, but no one who got low doses of the mTOR inhibitor even got a symptom of COVID-19. So this is encouraging that we're starting to hone in on the right clinical development endpoint for mTOR inhibitors to enhance immune function in older adults. And we want to test this with our next generation rapalogs. So this is just our pipeline at the company, our lead compound. We're going to test both to see if we could decrease severity of viral respiratory tract infections and in oncology. We have a backup compound also moving forward in these indications. We have a super rapalog moving forward for autoimmune disease, and we're also developing a topical rapalogs for dermatology indications. So just going to, I know we're running out of time. So we're just developing best-in-class rapalogs, and we're excited to see whether we can have some benefit for aging-related conditions in humans. And I'll stop there.
Thanks a lot, Joan. It's an excellent speech. Due to the time limit, so I think we can only take a maximum 2 questions. So first thing first, please.
Thank you very much, John, for your fantastic sharing. So I'm in the first in selections with Fuse Mueller, okay, in Switzerland. So for the mTOR deficiency, more than double lifespan in selections, okay. So my question is, first, have you repeated your TOR 101 and the others in selections for longevity study? Mouse or even like a pet dog to have the longevity effects and the first patients. The second question is whether you get to 101 is synthetic or modified from Rapamycin or is from natural products. And the third question is, I saw you are also in Basel or in Switzerland. So whether you also test the side effects, whatever, use the organoid or organ on chip for such side effects, whatever. Thank you very much.
Sure. So your first question, we haven't done longevity experiments with TOR 101, but I think it would be really interesting to look at, is a TORC1 inhibitor going to be better than a TORC1/2 inhibitor because of the data mainly from Dudley Lambing that TORC2 inhibition actually may decrease lifespan. C. elegans is probably the worst species to try to test longevity effects of rapalogs because it's tough to get rapalogs into C. elegans, and it may be because the bacteria in the food eat the rapalog before it gets, this is what I'm understanding from C. elegans people. You have to massively load the food with the rapalog to get it in. But I think we could look in flies and, even at the intervention testing program, look in mice. I think your second question was, is this a natural product or is it a chemically modified version of rapamycin or first generation rapalog? It's a chemically modified version, synthetic version of rapamycin. And then your third question, I'm not sure I understood.
Because transcribe, so in Russia, he made the platform for the organoid and the organ on chip testing faster for, especially for human side effects test. Okay, so whether you also have this kind of, because you still use the mouse, all right? So if for some side effects, okay, so if you use a human organoid, okay, or organ on chip.
So
you test for side effects for human.
Yeah. So to get into humans, the regulatory authorities like the FDA require you to use 2 species of animals. You can't use a chip yet. You have to use a rat and a dog, or a rat and a monkey, or a rat and a guinea pig, or a mouse and a guinea pig. There's like regulations that you have to use these species. So we've used a rat and a non-human primate. The non-human primate is probably the most interesting because it's obviously closer to humans, and we're seeing no tox. It's really remarkable. So it's going to be fascinating. It's likely that this isn't going to have much tox in humans either, because the toxicity that you see, for instance, with a everolimus in a rat, a lot of that tox you also will see in a human. So it's actually kind of predictive.
Yes, thank you very much.
Sure.
Thanks a lot. Anyone else like to ask? Maybe one more. Sorry.
Very impressive speech. I want to know whether talk 101 could use to healthy people and extend their healthy life.
Potentially. What's the most exciting about this compound is its safety. And you really have to have a safe drug to give to a healthy person to prevent disease, because there's no point in preventing disease if you cause disease. So this kind of profile is very exciting. If, we have to make sure it has this same safe profile in humans, but it's the kind of profile that you want. If you're treating a disease, you can put up with more side effects than if you're preventing disease.
Joan, great presentation. Thank you. I have a very quick question, maybe naive one. So you have this focus on immunological improvement. But what about sort of simple things like frailty score? Do you think that would not give you the better result?
Such a good question, Vera, because we may be, we just have a lot of clinical data with rapalogs and mTOR inhibitors from lessons learned from our previous trials. But there's some really interesting data that's been generated now of rapamycin in sarcopenia models, that the mouse data is actually a little bit better than the rat data, because the mouse, they can look at function, and as you know, old rats just kind of sit in their cage and you can't get them to move. But I think it's something we're likely to do is look at the effects of this rapalog on sarcopenia. And that is a beginning to more broadly looking at frailty. It's just like muscle is a little bit more specific, where we can look at size, fat infiltration, strength, twitch force.
Okay, thanks, Joan. It was an excellent talk, of course. And hopefully we will see you in person next time.
Yes, thank you so much. Thanks, everybody.